Hello, my dear students, welcome back. So, our second session on biotechnology. Okay, I welcome all of you back. I hope all of you are very alert after the examination, and now you have a quest for learning and asking doubts and getting or giving your maximum cooperation in online learning. Okay, so your biology exams, I hope all of you have written well. Then see, listen here. In the last class we have discussed what is an rDNA. Okay, today we are just starting with who has developed or who has produced the first rDNA. And I have given the name in the last class. Who are the scientists who developed the first rDNA? Tell me. Who remember it? Tell me. Yeah, it is Stanley Cohen and Herbert Boyer. Okay, so Cohen and Boyer developed the first rDNA. Okay, how they have made it? How they produce the first rDNA. Okay, so understand, Cohen and Boyer, they have taken antibiotic resistance gene. Keep in mind, this is not insulin gene. They have taken antibiotic resistance gene from one bacterium named Salmonella typhimurium. You know the bacterium, Salmonella typhi. The bacterium which causes typhoid. So once again, Cohen and Boyer isolated, or they have cut and taken antibiotic resistance gene from the bacterium Salmonella typhimurium. Okay, then how this became possible? How they have cut the DNA from this particular Salmonella typhi? How could they cut the DNA? Is with the help of certain enzyme, not with any scissors or scalpels or micro scalpel or nothing. They have used enzymes. These enzymes were discovered by other scientists. Okay. Anyway, using restriction enzymes. Okay. With the help of restriction enzymes come under the category endonucleases. Once again, using restriction endonucleases enzyme, Cohen and Boyer cut the antibiotic resistance gene from this bacterium. Now what they have in their hand? They have a small piece of DNA. What is that? It is the antibiotic resistance gene. Okay, in the second step, they have introduced this gene. What is that gene? Antibiotic resistant gene. From where it has taken? It has taken from Salmonella typhi plasmid. From the plasmid of Salmonella, not from the normal genome. Okay, from the plasmid of Salmonella typhi. Then they have introduced this gene into plasmid of the plasmid. This plasmid is taken separately and they have introduced into this plasmid. This cut piece of DNA, antibiotic resistant gene, then they have introduced to another plasmid. Plasmid is what? Extra chromosome of the bacteria. Okay, and this gene portion they have introduced here. Now what is it called? It is called the rDNA. This was the first rDNA. Did you get me? Cohen and Boyer cut and isolated antibiotic resistance genes from a native plasmid of bacterium Salmonella typhi. Okay, a native bacterium strain. And they have introduced this to the plasmid of another bacterium. And they produced the first rDNA like this. Okay, and not only the production, they have done the next step also. Then as a next step, they have introduced this rDNA into a bacterium, not Salmonella, but a similar strain bacterium, Escherichia coli. What happened? They introduced this to the Escherichia coli bacterium. Okay. This Escherichia coli is having its normal contents as such. Okay, normal genome will be there, everything will be there. But this Cohen and Boyer successfully introduced this rDNA into what? Into E. coli. So just familiarize certain terms here. First, in order to take this DNA, this is a plasmid. First, it is the plasmid of Salmonella typhi. Okay. From this, what is taken? Antibiotic resistance gene is taken. Okay. How it is taken? It is taken with the help of, it is isolated with the help of restriction endonuclease enzyme. No doubt, I think. Certain enzymes are used to cut this, a portion of this, and it is cut and taken out. Then from the plasmid of a native bacterium, that is what is Salmonella typhi. Introduce it into another plasmid. An introduction and joining is done with the help of the enzyme DNA ligase. Okay, when this, a portion of the plasmid is removed and the new portion is inserted, it requires the ligating enzyme or the gluing enzyme, that is DNA ligase. Okay. Now this is rDNA. Then it is delivered to the host. Okay, there are so many techniques to deliver it. We will discuss it. Okay, gene gun device is there, micro injection method is there. That all things we will discuss. Otherwise, some membrane treatment is there so that this plasmid can reach into the bacterial cell. Direct delivery also there. We will discuss all things one by one. Anyway, just keep in mind, this is our gene of interest, the gene which we need to multiply in the bacterial cell. So this is the gene of interest. Then what is the role of the plasmid here? The plasmid. What is the role of the plasmid here? Okay, tell me, what is the role of the plasmid? Why can't we directly introduce this to there? This is not directly introduced to the genome. Okay, there are so many problems in success rate. So it is found that this is a better choice. Okay. So if a plasmid is introduced to a bacterium, it is not showing the high degree of rejection. It is getting, the success rate is high. So understand, when this gene is introduced to the plasmid, plasmid is here acting as a vector. The term is vector. What is the role of plasmid here? Plasmid acts as a vector. What do you know about the term vector? You know mosquitoes act as vector. Okay, they act as vectors. They carry pathogens to our body. Dengue, that means so many diseases, malaria. Okay, this all diseases we get because the mosquitoes act as vector. They carry that and deliver to our body. They carry the pathogens and deliver to our body. Similarly, here vectors, that means the plasmids, carry the gene and deliver to the host cell. They act as a vector or carrier. Okay, simple concept, the same term we use, vectors. So, gene of interest, vector, then delivered to what? Bacterium cell means this is the host. Keep in mind these terms. Okay, that only about the rDNA technology. The first rDNA was produced by Cohen and Boyer using the plasmid of Salmonella typhi. They have isolated gene from Salmonella typhi, introduced to the plasmid, and this is used as a vector, then delivered to Escherichia coli bacterium cell, and they have successfully delivered and produced the copies. So that is about the first step in the first attempt in rDNA technology. Okay, if this is clear, we shall move to another small section, and the next session is a very big session, but we will just take the first part of that. Okay, shall we move to the next topic? It is tools in rDNA technology. Major tools in rDNA technology. Are we using scissors, okay, scalpels, okay, cutting devices, or okay, plowing devices? Nothing. We are using certain enzymes and molecules in rDNA technology. Organisms or molecules, or mainly enzymes, are the tools in rDNA technology. The first and most important tool we have already discussed, that is what is a restriction endonuclease enzyme. So the first tool in rDNA technology, restriction enzymes. The name is giving what idea? Restriction enzymes. This name give what ideas? It restricts something. Okay, it's creating some restriction. It's blocking. It's breaking, we can say. Okay, the restriction enzymes. Okay, see, for that we should know what sort of experiments discovered this. How people or human first came to know that there are restriction enzymes. Okay, that means this was a sudden discovery. By the time, in certain experiments, scientists noted that certain viruses, we can call bacteriophages, are unable to attack bacteria. Okay, usually what are bacteriophages? Viruses which can attack bacteria. These are the viruses. Okay, their host is bacteria. They attack bacteria and make disturbances there. Its own expression there and alter everything. Whereas scientists have noted that the bacteriophages, certain bacteriophages, are unable to attack bacteria. Its DNA, bacteriophage's DNA, is not allowed to express in bacteria. This is due to two major reasons they have found. Its DNA, the DNA of the bacteriophage should express on the bacteria. Then only this have an infection. During infection, bacteriophage DNA will show its expression here. Then this bacteria is infected. But it is found that this DNA is not able to express here due to two types of action. One is methylation of DNA. Due to methylation of DNA, bacteriophage cannot express here. And other one due to break or cutting of DNA. Restriction. Okay, that means it is found that during a study of expression of bacteriophages in bacteria, it is found that two types of enzymes can block this expression. Okay, the expression of bacteriophages on the bacteria can be blocked by, or its DNA can be blocked by two types of enzymes. One type is a methylating enzyme, other one is a restricting enzyme. A methylating enzyme will disturb the DNA of this by adding methyl groups. It is methylated. DNA is methylated, then it has no expression. And the second type of enzyme, what is we need now here, that is restricting enzyme. This type of enzyme is cutting the DNA. When the bacteriophage is attacking the bacteria, this type of enzyme goes and cut the DNA. It's broken to pieces so that it has no expression. Okay, so understand this. When bacteriophages express on bacteria, if restriction enzymes are there, means it cannot express because its DNA will be cut by restriction enzyme. So this is an important discovery. There are enzymes which can cut the DNA. Okay, so what is? There are enzymes which can cut the DNA. That enzymes are called restriction enzymes. These restriction enzymes are of two categories. Anyway, restriction enzymes come under the group nucleases. In the last classes, +1 classes, you have studied hydrolytic enzymes. Digestion process and all. Okay, hydrolytic enzymes you have learned: proteases, lipases, lipid digesting, okay, protein digesting, carbohydrates, nucleases, nucleic acid digesting enzymes, nucleases. And this nucleases, it comes under restriction enzymes belong to nucleases. These nucleases are of two types: exonucleases and endonucleases. Exonucleases and endonucleases. And from the term you can understand it. If the DNA is, if this is the DNA, if the enzyme cut and remove portions only from the terminal segments, terminal portion, end portions, that is exonucleases. If the restriction enzyme is able to cut the DNA only from the ends, the terminal ends, then it is coming to this category, exonucleases. It cut at the ends. Okay? Whereas, if the restriction enzyme is able to cut the DNA in between, it is known as endonucleases, restriction endonucleases. Which type of enzyme will be best beneficial for? So, our desired gene, it is not necessary that it should be at the ends. It can be at any place. So, we need this enzyme more. Okay, restriction endonuclease enzymes. So, what is a restriction endonuclease enzyme? A restriction enzyme which can cut the DNA from in between the sections or DNA strands in between are called restriction endonucleases. Okay, if it cut only from the ends, that are exonucleases. I think it's clear. Then, which was the first discovered restriction endonuclease enzyme? So, restriction enzyme come to the class group nucleases and endonucleases. It can make the cut in between. That is more useful for our genetic engineering works. Then, if was a restriction enzyme, which is the first restriction enzyme discovered by scientists? It is Hnd2. Okay. The restriction enzyme which was discovered for the first time by scientists is Hnd2. And see, another one, and this one is from Haemophilus influenza D strain, like that means we will discuss the naming criteria we will discuss, and it's based on the second example we will discuss. Okay, another important common example of restriction enzyme is EcoR1. You know, Escherichia coli is the most important experiment material, very common experimental material, and this is a restriction enzyme developed from Escherichia coli bacterium. So, let us discuss how this is named, how the restriction endonucleases are named. So, the first one discovered is this, and taking this as an example, we are studying or discussing how is the naming done. The first letter of the name of the enzyme represents the bacterium name, genus name, Escherichia. Name of the genus name of the bacterium. It is what? Genus name. I mean, the genus name first letter will be the first letter of the restriction enzyme's name. Then the second two letters, okay, represents the first two letters of the species name. Species name, first two letters. Okay, and this one indicate the strain, R strain, which strain it is. And this number indicate the order of extraction. Okay, I mean this is the first one extracted from E. coli. Okay, about the naming of the restriction enzyme. With the help of this example EcoR1. Okay, now itself you know that EcoR1 is an enzyme developed from Escherichia coli. The first letter indicate the first letter of the genus name, and the second two letters indicate the first two letters of the species name. Okay, the R indicate the R strain, which strain it is. Then the number, the Roman number indicate the in which order it is extracted, order of extraction. Restriction endonucleases can cut the DNA in between. Okay, Then when they cut the DNA, suppose this is a DNA, when EcoR1 is used to cut the DNA, where it cuts, where it makes cut. Is it possible to make cuts everywhere randomly? Impossible. Every restriction enzyme has a specific site to cut. Okay, where this EcoR1 is able to cut, that region is called its restriction site. Okay, its cloning site. So the restriction enzyme can cut only at specific portion. For example, EcoR1 is able to cut only at a region where the sequence is like this. Okay. Listen here. If in this DNA where a portion contains like this, where is the portion like this, there only EcoR1 can cut. So EcoR1's restriction site is this site, GAATTC. GAATTC, that means what is special about this site? This is the restriction site or recognition site or cloning site of EcoR1. I mean, this EcoR1 is able to cut the DNA only at this particular site. This is the cloning site, or it is also called restriction site of this particular enzyme. Every enzyme it is different. Okay, then what is special? When EcoR1 is used, this enzyme comes and make a cut here. Okay, between G and A. Here also, in between G and A, it makes cut. So, it is cutting the DNA like this. Okay, the DNA strands will separate. We will discuss with the example, with the diagram for the next class. Okay, with the help of a diagram, we will explain it in the next class. Now, just understand, every restriction enzyme has a particular recognition site. And very important thing, that recognition sites are palindromes. Palindromic DNA sites. Palindromes. Okay, what do you mean by palindromic DNA site? What is a palindrome? Which reads same on from left to right and right to left. Okay, for example, Malayalam is a palindrome, you know that. Okay, Malayalam, this side and left to this and this side to this, it reads same. So that is a palindrome. And a DNA palindrome means it should read same on same direction. From 5' to 3' direction on one strand and from 5' to 3' direction on the other strand. If it reads same, that is a DNA palindrome. So 5' to 3' here is GAATTC. Here also 5' to 3', it's in opposite direction, okay. 5' to 3' is GAATTC. So, a palindromic DNA is a DNA sequence which reads same, bases same in the same direction on both the strands. Okay, clear I think, simple fact. Only this kind of sequences these enzymes recognize. Not only EcoR1, all restriction enzymes make the cuttings possible only in the palindromic DNA sequences. These sequences are known as recognition sites or cloning sites or restriction sites of the enzyme. Okay, so which is the recognition site or the palindromic sequence of EcoR1? Simply don't write GAATTC, you should write both the strands. Okay, GAATTC, GAATTC, 5' to 3', 5' to 3'. Both the strands you have. This is the palindromic sequence of EcoR1. And your textbook is asking some more examples. You try to find it for the next class. Otherwise, if you are not able to get, I will give it. Okay, your textbook is not giving any other example for palindrome, but you have an exercise question. You have to find out 5 more palindromic sequences of enzymes. Okay, try to find it out for the next day. So keep in mind, try to find it out 5 more palindromic sequences of different enzymes. Then try and if okay, next class we will discuss it, and next class we will elaborate this ECOR1 action with the help of a diagram representation. Okay, this is a new concept for you. Study thoroughly at home. Watch the videos repeatedly and study thoroughly at home. Okay, thank you.